So, my goal is to describe to you a project that we have at Iowa State University, and the level at which I'm going to describe it, I want you to be able to walk out of here and be able to tell somebody else what it is. I'm not going to go into any great detail because, in fact, we've only run one experiment so far, and it just stopped this last week, so we're really very much at the beginning of being able to use this facility. So again, I just want you to know exactly what it is. So Enviratron was, for us, a different concept in how to do high throughput phenotyping, in that many facilities that are out there convey the plants to an imaging and sensors bank, and while the plants are there, all of the data are taken and then the plant gets put back into the environment. The primary difference between Enviratron and these other environments, or these other systems, is that rather than moving the plant around, Enviratron is moving the sensors around and the plants are staying in the same place. The reason that we wanted to try to do that is because we didn't want to introduce any kind of artifact in what the environment was for the plants. The plant continues to sit in one place and be in exactly the same environment. So let me show you what it looks like. So here's the facility, and I don't have a pointer, but hopefully you can follow this. So you see there 8 red boxes. You have a pointer? That's a different computer. You see 8 red boxes, and those boxes are the chambers. And inside of the chambers, the portion of each box that is toward the outside is where the plants are sitting. The portion of each box that is toward that red robot, the rover that wanders around in the Enviratron facility, that's an anteroom where the robot goes into the chamber and a curtain comes up so that the robot can image to see what's in the chamber and then decide which leaves that it wants to go to, which plants that it wants to convey the sensors to. So the part that's a little bit hard to see is what represents the tape on the floor. There are yellow dashed lines, and the robot goes into each of those. So if you want a closer up view and description of that image, you can see it at poster number 24, that Darwin Campbell, who's here, will be at that poster at the time that we have our poster session. So this is what the chamber looks like. On the left side there, you can see that curtain to the anteroom. The vestibule is on your far right. That vestibule is where the rover comes in. That screen that's got the red dots or red dashes around it comes up, and that's where the plants are. In the top image on the right, you see an aisle camera view, and so that's showing you sort of a fisheye lens view of if you were looking down among all of those 8 growth chambers that Percival built, what it looks like, and the robot is sitting there in the middle. The bottom right view, the chamber camera view, shows the vestibule at the lower half with the robot in it, and the plants on the upper half, and the robot has an arm, and that arm has lots of different sensors and is being taken to each of those leaves. Some of the problems that we've seen are that as the plant grows, the leaves change position. Around the leaves move some, and so we've had to do a lot of work on figuring out how we're going to find which leaf, whether we're going to be able to visit the same leaf with every pass. The answer is likely no. So it's been very interesting to figure out exactly what we should be trying to do and what the level of detail we can get at are. Certainly, it's been an interesting collaboration among engineers and data scientists and plant scientists, you name it, we've had them working on this project. The things that are controlled are shown on the left, and on the right is the range that is controlled. Whether each of the environmental variables that you see there are both controlled and measured, so we do have both what it is that we expect the chambers to be doing and measurements for what it is the chamber is actually doing. So we're controlling temperature, day length, light intensity, humidity, CO2, and soil moisture, sort of. So soil moisture, of course, we can add it. We can't take moisture out. The rover looks like this. So you see the robot there on the left, It has an articulated arm that has blue joints, and at the top is what we call a head, as if it were truly a robot walking around with all of its sensors on its head, just like all of your sensors are on your head. That head has a 3D imaging system, an RGB camera, thermal imaging, a fluorometer probe, a hyperspectral camera, a halogen line light, and a laser profilometer. I hope I said that right. So here's an image on the left of the rover visiting some plants in the growth chamber. And here are some pictures of plants under different temperatures from 22 Celsius up to 35 Celsius, where that middle temperature and those middle images come from maybe not an optimal, but a very good environment for maize growth. Here are some example images that we've collected. Of course, there's a soybean there on the left. We have some on the right that are maize, because if you live in Iowa, you work on corn and soybeans. We have a system where we can program the chambers, of course. So what you see in this example of that system is the experiment queue and status. So the first experiment is at the top. The experiment ID is shown there on the left. You see experiment IDs 1, 3, 15, etc. So if we go into one of those experiments, we can look at experiment detail, where we're putting down information about the plants and genotypes of those plants. There's a focus genotype, for example. And then we do some experimental setup to show how we want the lighting, temperature, humidity, etc. to be controlled. And here you can see that we filled out lighting through humidity, but CO2 and watering have not yet been programmed or are not monitored in this particular experiment. The interface where you adjust what your lighting and temperature looks like is a very modern interface. You can drag things around and decide how long you want your day lengths. You can also decide how you want your pots laid out based on the size of the pot, and you can number based on the size of the pot, and you can number the pots. For example, if the rover knows which pot you want to have measured at any given time, it can select them. And this is a list of tasks that are to be performed in each chamber. So once you get the whole thing put together, you may in fact want to look and just visualize whether in fact what it was that you put into the system is what both the chamber and the robot are programmed to do. So one of the things that's been very interesting in our discussions, the group of us that's working on Enviratron is trying to figure out, what do we do when we have a rover that's wandering around among 8 chambers? So we're going to get measurements that are each 15 minutes off from each other until we realized we could set all of the growth chambers 15 minutes apart, and then we're making simultaneous measurements, right? So there are all kinds of things about how we do the experimental setup that we could take measurements over time of chambers that are programmed to do the same thing. We can do measurements at exactly the same time in many different environments, so that there's a lot that can be controlled. So the at-a-glance overview of each chamber's activity shows, instead of how it's programmed to be functioning, what it is that it's doing. And for data management, our overall scheme is what's shown here. In the top left, you see the person who is designing their experiment. Their design goes into the Sciverse discovery environment, and that design becomes a part of the experiment sitting on the Sciverse data store. From the rover makes its measurements. We have a local server that, as the rover is functioning, it drops its information onto the local server, and then the server goes through the cloud and also ends up at the data store. We are now at the point where, instead of just making sure that we get all of the data that the rover is collecting and that the chamber is measuring, we can start doing some analysis. So I just want to go back to looking at this overall view. As I said, my goal was for you to be able to feel like you know exactly what Enviratron is and where we are as far as status with this project. So it's 8 growth chambers, and it has a number of different variables that we can modulate, and the idea is to control the environment more so than what it is that we have in other systems where plants are conveyed from one environment to another, and it's just now operational. So with that, I should point out that Darwin Campbell, who is at the back of the room, is here and probably is the most knowledgeable about the current status of Enviratron. You'll find him at the poster. Scott Zeracore is the programmer that has done a lot of the work to set up the infrastructure, the computational infrastructure, and Yin Bao is a student, now postdoc with Lee Tong, who has designed and put together the robot. Thomas Luberstedt, Steve Howell, and Steve Whittam have really been the people that have led the development of this project as far as being the biologists that are interested in seeing that we get this instrumentation up and going and useful. So with that, I'll either take a question or two or move on to the next speaker.